Okay, so here we go. So chapter 25 is all about fluid balance, managing electrolyte balance, as well as acid-base balance. Today we will talk about body fluid compartments, water balance, and we'll start electrolyte balance. We'll probably get through sodium, but we won't get through the other ones, and that's where we'll pick up at our next lecture. So for body water content, infants have the highest percentage of body water. This is because they have low body fat and low bone mass. And then adult males versus adult females, adult males will have about 60% of their body is water and about 50% for females. This is due to differences in body composition. Adipose tissue is the least hydrated tissue at 20% water. Muscle tissue is the most hydrated at 75% water. This content will decline with age, and this is due to change in body composition. Okay, so adult males are about 60%, adult females are about 50%. Adipose tissue is the least hydrated tissue, and skeletal muscle tissue is the most hydrated. There are two main fluid compartments. And depending on when you downloaded, if you downloaded this PowerPoint, there may have been a typo, which when I was going through, I realized that the math would not work. So 2/3 of body water is in the intracellular fluid. So we have several trillion cells, and there is cytosol. That cytosol is mainly water. And so if you add up all the volume of all those cells, that is intracellular fluid, and it makes up about 2/3 of total body fluid. Extracellular fluid is the remaining 1/3 of body fluid. It is comprised of two separate compartments: plasma, which makes up about 3 liters, and then interstitial fluid is this fluid between cells and between the cells and the plasma. And that's going to be the greatest proportion of extracellular fluid. There is also lymph, cerebrospinal fluid, humors of the eye, synovial fluid, serous fluid, and GI secretions. If you remember, okay, so just to make sure that we are all on the same page, we have intracellular fluid. And then we have the surrounding fluid. It's in between cells. It's between the cells and the plasma, and that is interstitial fluid. And then we have plasma. I will not be separating plasma and interstitial fluid really as separate compartments, because these two extracellular fluid compartments, they equilibrate quickly in most situations. But do you remember when we're talking about intracellular fluid versus extracellular fluid, that they are diffusing across these spaces and across these membranes? So this diagram is just showing how 2/3 of body water is intracellular, so within those 30 trillion or so, probably more cells, and then interstitial fluid plus plasma make up the extracellular fluid, and that is about 1/3 of total body water. Water is a universal solvent. If you go back to 1141 and you think about how there is oxygen, and then we have hydrogen coming off, and that creates a partially positive side and a partially negative side. So water is a polar molecule. By having these partial charges, it allows it to interact with the ions that are in solution, as well as if another molecule is also polar, it can work with that one. Water can pull molecules apart by opposite charges attract. So if a molecule has a negative side, then it can start to pull with the positive side, and the reverse it happens as well. Solutes are the substances dissolved in water. There are two classifications of solutes. The first are non-electrolytes. So non-electrolytes are most of them are organic molecules. They just do not dissociate in water. The prime example of a non-electrolyte is glucose. So if you take a beaker of water and you drop a sugar cube into the water, the same number of glucose molecules in the sugar cube will be the same number of molecules in solution. So it's a 1-to-1 ratio. If we take an electrolyte, these are substances that dissociate into ions in water. So if we now drop our salt cube and we have 1 millimole of salt, then we would end up with 1 millimole of sodium and 1 millimole of chloride. Okay, so the salt has dissociated into a sodium ion and a chloride ion. And the reason why this becomes important is because of osmotic power. So remember, we talked a little bit about osmolality and osmolarity. Just use those terms interchangeably. One is per liter of water, one is per kilogram of water, and 1 liter of water equals 1 kilogram at 4 degrees. So they're very, very interchangeable as far as terminology. Why is osmolarity important? Osmolarity is important because it affects the movement of water. If we have a semi-permeable membrane, like a plasma membrane, and we have water, and we put salt in one side and glucose in the other, then we would have twice as, and we start with the same concentration. We would have twice as many particles on the salt side as we would the glucose side, because it doesn't dissociate. And then water will move from its concentration gradient. So if you have a low solute, you have high water, the water will then move to equalize the osmotic pressure. So how many particles are on each side of the membrane by concentration. Electrolytes have a much greater ability to cause fluid shifts. So today's topic is fluid balance, and fluid balance is primarily going to be controlled by sodium. So non-electrolytes do not dissociate in water. An example is glucose. They do not contribute nearly as much to fluid balance as electrolytes. So electrolytes tend to be salts. They're going to dissociate into ions in water. So sodium chloride dissociates into a positive sodium ion, a negative chloride ion. And because they dissociate, they contribute more to the ability to move water in the body. So we have our cells, and then we have our plasma and the interstitial fluid. Okay, so this is extracellular fluid compartment, and this is the intracellular fluid compartment. Inside the cell, in this intracellular fluid compartment, the main cation is potassium. The main anion is phosphate. Why is this true? So for potassium, the reason is going to be that sodium potassium ATP pump. And then this has to do more with proteins. But that sodium potassium ATPase pump that is in every membrane and is always churning is going to establish the cation distribution. So with that sodium potassium pump, we get 3 sodium out and 2 potassium in. Then potassium can somewhat equilibrate through leach channels, but we definitely get a buildup of sodium in the extracellular fluid. And then to balance sodium, chloride follows. There are channels for chloride as well. So you do need to know the major cation and the major anion for extracellular fluid. So that would be sodium chloride that's separated into anions, but it does kind of make it easier to remember. If you think of salt is going to be a major influence in the plasma for maintaining things like blood pressure. And then intracellularly, the major cation is potassium and the major anion is phosphate. So we have our fluid compartments. We've talked about how electrolytes are the main contributors, so ions to fluid balance. And now we're talking about the major players for each compartment. This is a graphic of what I just stated. If you remember, then we have extracellular fluid is this one with plasma and interstitial. They're very similar. So we usually just talk about extracellular fluid versus intracellular fluid. Extracellular fluid, the main cation is sodium. The main anion is chloride. Then if we go to intracellular fluid, the main cation is potassium, and the main anion is going to be phosphate. Negatively charged proteins are also going to contribute to osmotic balance intracellularly. Here we have osmotic and hydrostatic pressures again. Osmotic pressure is the pull. It's this, we call it the suck. It draws water back into either the cell or into the plasma. Okay, there's very little proteins in interstitial fluid. So for this one, we do just kind of talk about plasma versus intracellular. They both have proteins. So in the plasma, we have albumin, it's a plasma protein, and it pulls water into the plasma. In the cell, we have a lot of proteins. We have ribosomes, we have protein products, we've got proteins inserted here, there, kind of in everywhere. And so those proteins are going to pull to keep water in the cell. So we've got proteins that are creating osmotic pressure, and then we have hydrostatic pressure. And the main hydrostatic pressure is arterial blood pressure. That's going to push fluid into out of the plasma into the interstitial fluid where it can equilibrate. There's not a lot of hydrostatic pressure in the interstitial fluid because it gets reabsorbed in that capillary. So 2 pressures though, osmotic is the pull and hydrostatic is the push. This is going to create a continuous exchange and mixing of fluid. Water moves freely along its osmotic gradients. Water sometimes can move paracellularly between cells if the junctions allow, or it can use aquaporins. But it is considered that water can move pretty freely in and out of cells, in and out of this interstitial fluid, in and out of plasma. It pretty much is going to just follow that osmotic gradient. A change in solute concentration of any compartment leads to net water flow. So if we can increase the amount of salt in the plasma as compared to intracellularly, then water will flow from a high concentration of water to a lower concentration of water. So by changing the solute concentration, which in most cases is sodium, then water will follow. So water tends to follow sodium. So if we regulate sodium, we regulate water. It's not an all or none, but they do have that relationship. Substances must pass through the plasma and the interstitial fluid before it can enter the intracellular fluid. And then exchanges between the interstitial fluid and the intracellular fluid occur across the cell membrane. For some substrates, like nutrients, waste, and gases, that flow is going to be unidirectional. And for water and ions, it's going to be bidirectional. And the next graphic is really nice to display this. We have gases that are unidirectional, meaning that they flow from the extracellular fluid to the intracellular fluid, or they can flow from the intracellular compartment to the extracellular. So it's an either or. They're flowing in, they're flowing out. And the same occurs with nutrients and waste. So these four are unidirectional. When we get to water and ions, they are going to be bidirectional. So ions, remember, if you remember, like with sodium, we are going to use that concentration gradient to co-transport things into cells. And then we have that ATP pump that pumps sodium back out. So sodium goes into cells, and then it gets pumped back out of cells. And as we just talked about, water follows salt. So water is going to go into cells, and then it's going to go out of cells. So water and ions move in both directions. And that is really what fluid balance is. It's balancing the concentration of water and ions between compartments. Okay, so now we're going to move on to osmolarity. Water intake must equal water output. Water intake is usually in the form of beverages. There's also some water in food. And then this metabolic water comes about from oxidative metabolism, where you take glucose, you break it apart, and if you use oxidative metabolism, it breaks into carbon dioxide and water. So that's where we get a little bit of this water. And you can change these amounts. The one that we tend, if we need more water, we tend to increase our fluid intake. And this is done by something called the thirst reflex. For our average output, we'll talk about sensible versus insensible water loss. So feces and sweat can greatly increase if we're exercising, so this one can change. Insensible water loss doesn't change a whole lot. And then our urine output can change quite a bit. So if we go out and we forget to take our water bottle with us, and we don't take in enough water, and we start to get dehydrated, we can decrease the urine amount to about 500 mils. So that liter, that's left can be reabsorbed into the body to maintain plasma volume and osmolality. If, on the other hand, we are being very good and remembering to drink lots and lots of water, then we can increase that urine output to match. But in general, the intake should match the output. So regulation of water intake, as I mentioned, is controlled by the hypothalamic thirst center. There are osmoreceptors. So right now, okay, this graphic gets a little ahead of itself because sodium's going to have two roles. It's going to have a role in maintaining osmolarity and a role in maintaining plasma volume. So this graphic will come back to it. But for now, we're only going to talk about this half, okay, because we're talking about the thirst response. If osmoreceptors in the hypothalamus sense that there is a increase in osmolality, so an increase in osmolality means that there is too much, too much salt present for the volume of water. So osmolality is a concentration. It's how much salt do we have in plasma. When we look at this left side, we're looking at the effect of osmolality or osmolarity on water balance. Because if we don't maintain proper osmolality, cells shrink or they swell. If you go back and you kind of remember a little bit about tonicity from last semester, if we put cells in you could put them in 3 beakers. You could put them in hyper, iso, and hypotonic solutions. And we took a red blood cell, and in isotonic, it stayed that same shape. In hypotonic, does anyone remember if the cell, did it swell or did it shrink? In a hypotonic solution, the red blood cell, it swelled up because the water moved from high water, low salt into the red blood cell. In hyper, it shrunk, okay? So it crenated. So this tonicity of the solution affected the red blood cell shape. And when we talk about osmolality, we're now talking about this on a system-wide scale. If our blood is too salty, so it's going to be hypertonic, then water moves out of our cells to try and equilibrate that osmolarity, because water has no choice. It has to move along its concentration gradient. So if our osmolarity increases, like up here, then if we didn't do any adjustment, then the water would come out of our cells and our cells would start to shrink. And our cells don't like to change shape. They prefer to stay a constant size. In general, like swelling sometimes can be more of an issue, like if you get cerebral edema because we've got our skull and we only have so much space. But regardless, our cells want to maintain this isotonic condition. So osmoreceptors, they're just like any other cell in your body, and that they're susceptible to the tonicity of the fluid that they're in. The way that they're different is that they get to, when they start to shrink, then they are going to send action potentials to the hypothalamus. So all the cells are experiencing roughly the same effect, but osmoreceptors have a direct connection to the hypothalamus. So as they start to shrink, they will start firing off action potentials to the hypothalamic thirst center, which then stimulates the sensation of thirst. We're like, oh, I think I need to go grab something to drink. We ingest water, and then the water moistens the lining of the mouth, throat, and stomach. And it is going to tell the hypothalamus we're good. Because if we waited for the actual osmotic balance to get back to completely like to normal, then we would have probably over ingested water. So this is just a thing like, okay, I think we're good. Let's slow down and take a break. And if we need more water, we'll come back. But if not, then we're good. Then water gets absorbed in the GI tract, increasing, and then it transfers into the plasma, increasing plasma volume, decreasing osmolality, which then feeds back. For water loss, we have some what are called obligatory water losses. And these are water loss that is considered unavoidable. We have insensible and sensible. So sensible means you can actually feel it. So you can sense the presence of that water loss. The best examples are sweat. So when we are sweating, we can see those drops running off of our forehead. The other good example of sensible water loss is the loss of urine. So you can tell when you are losing water due to urine release, excretion. Insensible water loss is water loss that you cannot feel. So we lose, when air comes into our lungs, we humidify it. So we increase water vapor. Then when we exhale, expire that air, some of that is recaptured, but a lot of it is released back into the air. So if you think about like when it's really cold and you're breathing out and you can see your breath, that's your breath like crystallizing in the air. So then I guess we could perceive it, but under most conditions, we don't. So loss of water vapor from respiration, the lungs, is considered insensible. So you do need to know the difference between insensible and sensible water loss, as well as examples of these terms. So now we're going to look at the influence of antidiuretic hormone on water balance, and then we'll look at the effect of aldosterone. So the posterior pituitary is going to be responsible for secretion of antidiuretic hormone. So if the pituitary increases secretion of antidiuretic hormone, then those aquaporins get, if you remember from urinary system, we have our proximal, we've got our loop, we've got our distal convoluted tubule, and then we have our collecting duct. At its most concentrated, filtrate is at 1200 milliosmol. Once it gets to the distal convoluted tubule, it is 100 milliosmol. And then it is going to go to the collecting duct to be excreted. If we are dehydrated and need to reabsorb water, so high ADH, then ADH is going to place aquaporins in the collecting duct so that water can be reabsorbed. So it leaves the collecting duct and then it enters the vasa recta for reabsorption. So high ADH increases the number of aquaporins. It increases the water reabsorption, and it really should be from the collecting ducts because water leaves the collecting ducts and then is reabsorbed by the vasa recta. Leads to concentrated urine, and that water that has been reabsorbed is going to increase the volume of body fluids. Those hypothalamic osmoreceptors sense that we have a balance in the force and will then adjust the amount of ADH that is being released by the posterior pituitary. So ADH is more involved in water balance. It's the movement of water out of the collecting duct so that it can be reabsorbed to reestablish osmolarity. So ADH is more involved in osmotic regulation. In regulating water balance, this is a graphic of what we just talked about, but it's starting to bring up again. So I'm going to go ahead and go over to the whiteboard. We look at how sodium is going to affect osmolarity, also known as like the ratio of water and salt. Okay, so it's a concentration. The reason why we're concerned with that is because water follows its gradient. And in very simplistic terms, cells will end up shrinking or swelling. So that's a concentration issue. The factors that are going to control that ratio, that concentration, are going to be osmoreceptors in the hypothalamus, which then affects the thirst reflex, as well as ADH release. Thirst reflex and ADH release, both of these, if you were to promote the thirst reflex and increase ADH, both of these would promote an increase in, we'll call it extracellular fluid water. The thirst reflex would do this by you ingest water, eventually gets taken up by the small intestine, and then it will get reabsorbed into the plasma, extracellular fluid. ADH reabsorbs water from the collecting duct, and then it goes from the vasa rectus into the extracellular fluid compartment. We also need to be concerned about total body salt content, because total body salt content is going to affect extracellular fluid volume and therefore blood pressure. And when we talk about extracellular fluid volume, we're mainly talking about blood volume. People are like, you might be like, so aren't they the same? But they're not, because, okay, so let's say you like chocolate chip ice cream and you received a carton of chocolate chip ice cream, but it was only half full. It would have could have the same concentration of chocolate chips between so we get a carton, we open it, it's half full, But the concentration, the number of chocolate chips in the vanilla ice cream, is the same as if you open up a carton that is completely full. So the ratio to chocolate chips to vanilla ice cream is the same. So these have the same osmolarity. But if you are thinking now about blood volume, is your body going to be okay with 3 liters of blood flowing through your system versus 6 liters of blood flowing through your system? You need this one to survive. So we need to maintain osmolarity so that our cells aren't always shrinking and expanding, and the water balance in between the cells and extracellular stays good. But with total body salt content, we're going to have the correct volume of correct osmotic fluid. So total body salt content is going to be what controls having the right amount of blood volume, because that's what we need for blood pressure. Total body salt content is going to be, because where salt goes, water follows, so we get blood volume and blood pressure. This is going to be under baroreceptor control. And then it also is going to feed into that renin-angiotensin-aldosterone system. Again, we'll talk about this a little, this is like a little bit later in the lecture, and the role that aldosterone plays. And that's what this graphic is showing, that on this side, we're looking more at maintaining osmotic balance. So the osmoreceptors are going to feel, they can sense that the extracellular fluid osmolarity or osmolality is increased. That is going to stimulate the posterior pituitary to release ADH, antidiuretic hormone, which targets the collecting ducts of the kidneys to insert more aquaporins. That allows water reabsorption by the vasa recta, and it will return it to the extracellular fluid to return osmolality to normal. If we are looking at total plasma volume, so that other part, then if there is a decrease in plasma volume, the baroreceptors are not getting stretched, and then that is going to target the renin-angiotensin-aldosterone system, which we'll go back through in a second. Okay. When we're trying to maintain osmotic balance, the main factor is, the main sensors are going to be osmoreceptors in the hypothalamus. The main response is going to be ADH release, which is going to result in the reabsorption of water. Then we have a couple of disorders of water balance. Dehydration is when we have extracellular fluid water loss due to hemorrhage, severe burns, profuse sweating, prolonged vomiting. This one also involves electrolyte issues. Hypotonic, when there is cellular overhydration. Sometimes this occurs with rapid excess water ingestion, and kind of counterintuitively, this is like, not that this happens a lot, but the times it sometimes will happen is with marathon runners when they rehydrate, because they know that they've like, the thirst response is for like, oh, I've been out jogging and I've lost some water, I need to like quench my thirst. But marathon runners are pretty aware that they've lost a substantial amount of fluid, and so they will go above and beyond what their thirst response is, because they're like, well, I need to rehydrate. But if they overhydrate, then their cells can go hypotonic. This is Dehydrate, cells shrink. Overhydrate, cells swell. Edema is atypical accumulation of interstitial fluid. So normally the extracellular fluid, the interstitial fluid, two components of the extracellular fluid, they're usually in balance. But sometimes there is accumulation in the interstitial fluid. This is usually due to incompetent venous valves, so compromising venous return, sometimes congestive heart failure, an increase in hydrostatic pressure, which would be high blood pressure, sometimes inflammation. But edema is categorized by atypical accumulation of interstitial fluid. Sometimes it's also due to if like lymphatics have been removed, because lymphatics do contribute to fluid balance a little bit as well. Okay. So now we are moving on to electrolyte balance. We talked about how water moves because of ingestion, so bringing water in, as well as reabsorbing water from urine, so affecting those two components. Now we are going to look at controlling electrolytes. And you guys can probably somewhat guess some of the ones that we're going to talk about. The major extracellular cation is what? So it has a positive charge in the extracellular fluid. What is that ion?
Salt.
Very good. Okay, so we're going to talk about salt regulation. What is the major cation in intracellular fluid? Has to do with a pump.
Potassium.
Yep. Okay. So we're going to talk about sodium regulation. We're going to talk about potassium regulation. We're also going to talk about calcium because it's a major component in our bones, but also it's very important in action potential generation in neurons and skeletal muscle, and then in contraction in skeletal muscle. Why is sodium so important? It is the major extracellular, so this is plasma. It is the main ion in plasma. But aren't there other things? Kind of, sort of. So the concentration, the osmotic concentration in plasma is 300 millimolar. And this should look familiar because this is also the concentration in filtrate for glomerular filtration. Salt is responsible for 280 of them. So if you want to maintain balance in the plasma, you have to regulate salt. So the regulation of salt drives a lot. Okay, so electrolyte balance, we usually refer to it as salt balance because it's 280 of the 300. So it's the major player. So even though there are other electrolytes, acids, bases, and some charged proteins, salt is going to control the fluid movement. It is going to provide minerals for excitability, secretory activity, and membrane permeability. Right now, we're really focusing on fluid movement. Before, we've talked about how salt is also used with that ion gradient for co-transport, things like that. Salt enters the body by ingestion and metabolism. So our body needs 500 milligrams of salt, and usually sodium chloride, but of sodium per day. Our ingestion of salt is 5,000 milligrams of sodium per day. So we get plenty of salt ingested, which means we need to usually excrete quite a bit. Salt is lost in perspiration, feces, urine, and vomit. As I've said a few times, sodium is the most abundant cation in the extracellular fluid. It contributes 280 milliosmoles of the 300 milliosmoles of extracellular solute. It is the only cation exerting significant osmotic pressure. And that's because it's got such a lion's share of the osmotic balance that it is going to be the one that controls osmotic pressure. So sodium also controls extracellular volume and water distribution because water follows salt. And water doesn't have any option other than to follow salt because it is going to move by osmosis. And osmosis always flows from high to low concentration. So water is always, I mean, there are a few places where water can't move, right? So collecting duct, you can move or you can't move, or same with like the ascending limb. But in general, in most tissues, water flows freely and will follow salt. Therefore, changes in sodium levels, so the sodium amount of total body sodium, is going to affect plasma volume, and plasma volume is going to be the main player in blood pressure. Sodium content in the body changes, but extracellular fluid concentration remains stable because of movement of water in and out of the intracellular compartment, as well as ADH and thirst. So we change the water balance. This is what I was drawing on the whiteboard. When we are looking at sodium regulation, there are two pathways. They do work with each other. You know, they're not going to work against each other, and they do have some components in common. But for sodium concentration in the extracellular fluid, so you can think of plasma sodium concentration, that is the osmolality. So how 300 milliosmoles, which for sodium is 300 millimolar, that's a concentration of salt in plasma water. The sensor for maintaining 300 milliosmol are the hypothalamic osmoreceptors. If we get an increase in osmolality, meaning that there's too much salt present or not enough water present, then we are going to trigger the osmoreceptors. The osmoreceptors then in the hypothalamus tell the posterior pituitary to increase antidiuretic hormone, increased thirst mechanisms. This one increases water intake, and this one increases water reabsorption. And then that will return osmolality to 300 millimolar. With total body sodium content, it's going to be used to regulate blood volume and thus blood pressure. If blood pressure, so if blood volume falls, blood pressure falls. It's like putting water through a hose. If you don't have a lot of water, it's trickling out the other end, and there's not much pressure in the system. If you have a lot of water, sometimes you'll even see that hose, especially in the old-timey hoses, where it would bulge. And so if you think about putting a lot of blood through your blood vessels, they can stretch. And that is the stretch of the baroreceptors. So if we have a decrease in blood volume, a decrease in blood pressure, these are not getting stretched. It's the trickling hose. When we have low blood pressure, we also have a low glomerular filtration rate. because remember, glomerular filtration is directly related to hydrostatic. We'll come back here. So if we have a decrease in blood volume, that leads to a decrease in blood pressure and a decrease in baroreceptor stretch. If the blood pressure is low, we have a decrease. Whoops. Need a little bit more room. Decrease in glomerular filtration rate. And that is because hydrostatic pressure drives the amount of filtrate that is produced. You decrease blood pressure, you decrease hydrostatic pressure, you decrease glomerular filtration rate. You decrease glomerular filtration rate, you increase sodium reabsorption by the tubules because it's slow. It has a lot of time to pull the sodium back out. When you increase sodium reabsorption, you end up with low sodium at the distal convoluted tubule. So by the time you get back, you go through the loop of Henle and you're coming back up, the filtrate concentration is low. Then we get to that juxtaglomerular apparatus or complex, and it has macula densa cells. The macula densa cells sense that there's low sodium. And so it's like, that's kind of interesting. So then it tells the granular cells of the afferent arteriole to release renin. Renin, then it's going to take angiotensinogen to make angiotensin 1. It sends that to the lungs to become angiotensin 2. Angiotensin 2 is part of this here. So we get that cascade, we get angiotensin 2. And then that is going to have those 4 effects of angiotensin 2. One of those is going to be to increase aldosterone. So earlier today, we talked about this half. And then we had already talked a little bit about the renin, angiotensin, aldosterone system when we talked about blood pressure. So we're going to continue to talk about this part because right now we're talking about regulating sodium. So how much sodium do we want in our body? If we want a lot of sodium, or more sodium, then we will activate the renin, angiotensin, aldosterone system. There are no receptors that actually monitor sodium levels. Everything measures stretch or osmotic balance, but it's not like there's not like a technical sodium receptor or that monitors sodium. So what it does look at is it looks at osmolarity or it looks at stretch. Sodium water balance is linked to blood pressure, and that's because it affects the amount of blood volume, so the amount of water going through the hose. Changes in blood pressure or volume trigger neural and hormonal controls to regulate sodium content. Total body sodium content is indirectly monitored by baroreceptors because if we have a lot of salt, water follows increases plasma volume, increases blood pressure, and that can be sensed by baroreceptors. In contrast, when we're looking for concentration, osmolarity, those are osmoreceptors that we talked about previously. Okay, so now we're going to look at regulating sodium, not water. ADH regulates water. Thirst regulates water. Now we're going to look at baroreceptors and renin-angiotensin-aldosterone system to measure total salt, therefore plasma volume, and then plasma or blood pressure. Regardless of hormonal control, there is this obligatory reabsorption. Our body knows that we need to maintain a certain volume of salt to maintain a certain volume of plasma in our body. So regardless of anything else that's going on, 65% of sodium is going to be reabsorbed in the proximal tubules. 25% is going to be reclaimed. And we don't always say reabsorbed because it's also used for that osmotic medullary gradient. So, but 25% is reclaimed in the nephron loops. Sodium is never secreted into the filtrate. But so if you do the math, 90% of sodium is obligatory reabsorption or reuse. Otherwise, we wouldn't be able to maintain plasma volume kind of under any circumstances. So then we're going to look at what happens to the other 10%. When aldosterone concentrations are high, so there's a decrease in blood pressure, affects baroreceptors, and then it also affects those maculodensa cells. Renin gets released, we get to angiotensin II. Okay. One of the effects of angiotensin II is to promote aldosterone secretion by the adrenal cortex. Okay. So when we say aldosterone concentrations are high, this renin-angiotensin-aldosterone system has been activated. So decreased blood pressure, usually due to a decrease in blood volume. It's going to cause sodium to be actively reabsorbed in the distal convoluted tubule. And I'm sorry, this should be collecting duct. And water follows, extracellular volume increases. So then we get an increase in blood pressure as well. The reverse happens if aldosterone conditions are low. I kind of like to just do one side though, so it doesn't get so confusing. And with the renin-angiotensin-aldosterone system, we're usually talking about a decrease in blood pressure. The renin-angiotensin-aldosterone system or mechanism is the main trigger for aldosterone release. This is, in words, kind of what we've already said, as far as if we get a decrease in blood volume, that leads to a decrease in blood pressure. That leads to a decrease in GFR and an increase in sodium reabsorption at the proximal convoluted tubule, distal convoluted tubule. And that's due to things are going by so slow. This is sensed by those maculodensa cells, which then triggers the granular cells to release renin. Renin catalyzes the reaction to create angiotensin 1, goes to the lungs, it's converted to angiotensin 2. So that was that top half, sometimes of that graphic. Then we get to angiotensin 2 and its effects. For angiotensin 2, one of its effects is that it stimulates the adrenal cortex to release aldosterone. Aldosterone increases or stimulates the kidneys to increase sodium reabsorption. We're not going to talk about potassium secretion right now, and that will return the blood volume and then the blood pressure back to normal. Okay, so again, something you need to probably diagram out a few times on your own to remember its effects. Aldosterone is going to increase blood pressure. So increase aldosterone, increase blood pressure. Well, your heart says most of the time that's really good, but sometimes the blood pressure gets too high and could actually start to damage the heart cells. So we have our atria, and they produce atrial natriuretic peptide. When the atrial cells get stretched, so remember, ventricular cells are always getting stretched as they filled. So this would not be a good place to have this sensor. But the atria usually have most of its passive filling and then it contracts. So if the atria are getting stretched, that's a signal that blood pressure is too high. And so it will work to protect the whole heart by secreting atrial natriuretic peptide, which then overrides every other response. And it is going to inhibit antidiuretic hormone, inhibit renin, and inhibit aldosterone production. So it inhibits everything that's working against it. It's got that ultimate control. This will end up increasing excretion of sodium and water to decrease extracellular fluid volume and decrease blood pressure. So atrial natriuretic peptide, its effect, its end effect is going to be to decrease blood pressure by decreasing blood volume. This goes over if an increase in blood pressure increases the stretch of the atria, it releases ANP. And then ANP, it is going to inhibit pretty much everything. It inhibits the granular cells. It inhibits the adrenal cortex. And it is going to inhibit the release of ADH from the posterior pituitary. The end result is decrease in sodium and water reabsorption, decrease blood volume to decrease blood pressure. I'm not really going to go into the influence of other hormones. Estrogens, they act kind of like aldosterone. Progesterone blocks aldosterone, and then glucocorticoids promote edema. So they're there. They're not going to be on the exam, and I'm not really going to take the time to go into them. So I like this graphic better than the previous slide because it goes again through if blood pressure is falling, then there's a decrease in sodium filtrate because there's more time to reabsorb that sodium. This low sodium chloride concentration is going to tell the granular cells to release renin. Renin catalyzes angiotensinogen to angiotensin 1, goes to the lungs. ACE converts it to angiotensin 2. Angiotensin 2 has actually 4 effects. One is it will stimulate the thirst reflex. That's the one that's not on here. It is also a vasoconstrictor, and it stimulates the cortex to secrete aldosterone, and it will also stimulate additional ADH release. So this is just going over those 4 effects of angiotensin 2 and how it returns blood volume to then results in an increase in blood volume and an increase in blood pressure. Okay, so that is where we stop today. We'll pick up with potassium.